Hi, good afternoon, everyone. My name is Alicia Masonette, and I'm the Program Assistant for the Center for Pancreatic Cancer Research. And I worked alongside with Vicki to help organize the summer seminar series. Today's seminar speaker is Dr. Sheng Kai. He's an assistant attending in the Department of Medicine's Leukemia Service and also a member in the Ross Levine Lab. As a board-certified medical oncologist and physician scientist, he specializes in caring for patients with hematologic malignancies, including acute and chronic leukemias, myelodysplastic syndrome, and myeloproliferative neoplasms. Most of his time is spent performing laboratory-based research with the central aim of developing and investigating the mutations that cause acute myeloid leukemia and mechanisms of action, novel leukemia-directed targeted therapies. He hopes that his research efforts will ultimately result in a deeper understanding of how leukemias develop, as well as improve clinical outcomes for patients with blood cancers. Again, please welcome Dr. Kai.
Okay, so sorry about the delay there. I had a little bit of a glitch, but thank you for the introduction, Alicia. Here is my Twitter handle if you want to tweet questions at me after the talk, and Alicia gave my introduction. So I'm especially excited about talking with you guys because I was right where you guys are at in high school, where my personal career path to becoming a physician scientist started. So it was right around this time as a sophomore in high school where I applied for one of these summer research programs at the University of Cincinnati. And I got assigned to work with a neuroscientist who was trying to study kind of biochemical effects of damage caused by bleeding strokes in the brain. So when bleeding happens in the brain, what biochemical processes cause toxicity to nervous tissue? And we did this in pig models, using pigs as a model organism first doing our research. And I worked very closely with a postdoctoral researcher from China who actually was a practicing neurosurgeon when she was there. And so I just saw her do these amazing surgeries with her hands on these pigs. And then I was doing these biochemical assays. So it was the first great entry to me to say, wow, this is really cool, doing research on, on animals that might potentially be affecting and improving lives for patients that are suffering from strokes. And so that was my first time that I got bit by the research bug. The following summer, I played in an orchestra, and one of my friends in the orchestra said, hey, my dad is a researcher down at University of Cincinnati, and he's wondering if you might want to come. He's looking for people to come and work for in his lab in the summer. Would you be interested in doing that? And I said, sure. And the following summer, I went and worked with this guy, Dr. David Huey, who's a lipidologist, studies how cholesterol gets brought to tissues and gets transported back to the liver to get excreted. And so with studying that, mechanisms of kind of GI cholesterol homeostasis and how that potentially could be protective for heart disease. And so I was doing RT-PCRs, so looking at expression of this particular cholesterol receptor. And my very last week of that summer working with him, I ended up finding one specific band from one specific piece of tissue that was different from all the other pieces of intestinal tissue. And he said, well, that's interesting. If you want, you can come back the following summer and kind of continue to work this up. And so I did that. And we completed a project. And ultimately, the paper there was a paper that was published in a small journal. It was really fulfilling to kind of see a project go from its inception all the way to a publication. And so at that time, I was applying for colleges, and I was trying to decide, because I was trying to decide between going to a traditional 4-year university, or in Ohio, there was a medical university called Northeastern Ohio University's College of Medicine that had a kind of a guaranteed track toward med school. So it was a 2-year bachelor's followed by 4 years of MD. And from high school, you have a guaranteed admission into medical school. And so I was trying to decide between a guaranteed admission into medical school or going to a 4-year college and then having to apply for medical school then. At that time, it seemed pretty appealing to me to have a guarantee into medical school. But after having worked with Dr. Huey, I realized that I didn't want to give up potential of maybe doing more research. And so I went to Duke and I asked Dr. Hugh, I said, well, who do you think I should work for at Duke? He said, well, you should work for this guy. This is Dr. Bob Lefkowitz. He's a physician scientist. He's an MD physician scientist, and here he is holding his Nobel Prize that he won in 2012 in chemistry for discovering this whole family of what's called G protein coupled receptors. He figured out that there was this whole superfamily of genes that encode this receptor that there are thousands of these genes within mammalian cells and humans. And that the range of receptors, the range of ligands for these receptors go from anywhere as tiny as a single photon of light. So the rods and cones in your eye that allows you to see and perceive color and light are encoded by receptors for photons that perceive these things. And so from ligands as small as a photon to the adrenaline system that gives you the fight or flight response to so many other things in your body are involved or are regulated by these what's called G protein coupled receptors. And so I came and worked with him and worked on signal transduction pathways downstream of this receptor. We were transfecting cells and doing Western blots and mutating genes and putting this in, and I thought it was really cool. One of the cool things I learned from working with him was, the excitement of designing an experiment and getting a Western blot result that's literally coming out of the film developer and seeing Mother Nature reveal a secret to you for the first time. And that was when I realized, wow, I can't just go to MD and medical school. I want to pursue research. And so the next step in my career, I was trying to decide, MD or PhD. There are MDs who do research. There are PhDs who do research. And so for those of us who are undecided and still can't make a decision, you can do both. And you should know that there are federally funded programs that will pay you a stipend. So basically all of med school is free, PhD is free, and they pay you an annual stipend on top of that to be a kind of a physician scientist, a medical scientist, to go into these combined MD PhD programs. And so I went to Washington University in St. Louis, Missouri, chose to get my PhD with this guy, Dr. Tim Lay. And he is also a physician scientist, an MD trained physician scientist who's a hematologist by clinical training and also a leukemia biologist, a leukemia genomicist in terms of his research interest. And at the time, his lab was split up into two parts. One was studying kind of how T cells and natural killer cells kill cancer cells and virus-infected cells. And the other part of his lab was studying kind of leukemia genetics, leukemia genomics. And when I joined his lab, I worked on the T cell side and trying to study how T cells kill, but I was exposed to this field of leukemia biology, which I thought as an MD PhD, I thought, wow, these are some really interesting biological questions that can be framed in a clinical context that can directly impact patient care. And so that was a great training experience for me to see that through the lens of this particular mentor. So then I knew that I wanted to do cancer as a subspecialty in medicine. And so I wanted to first, before doing a cancer subspecialty, you have to be trained in internal medicine. And so I matched into the internal medicine residency program here at Cornell. This is a medical research track. So it's a physician scientist, what's called a short track. So in general, the internal medicine residency is 2 years long. It's 3 years long. But for the physician scientist track, they shave off a year here and add it onto your fellowship year, because if you're a lab track, they try to give you more time to spend in the lab. So I was matched in at the same time that I matched into the Cornell internal medicine residency program. I also matched into the oncology fellowship program at Sloan Kettering. And when I matched there, I worked with this guy, Dr. Scott Armstrong. He's an MD PhD physician scientist, and he was at Sloan Kettering at the time. And he is a pioneer in the field of a specific type of leukemia that kids get, in particular infants get, called MLL rearranged leukemias. There's a particular gene called MLL, and it can swap its pieces of DNA with other genes, a bunch of different genes, fuses together and creates a very potent oncogene that can cause leukemias in kids and also some adults. And And so he eventually left New York City to go back to Boston to become the chief of pediatric oncology at Dana-Farber Cancer Institute. But then I continued my research with this guy, so Dr. Ross Levine, who you met on Monday, who kicked off this whole seminar series. And again, Ross is a physician scientist with an MD training and also one of the pioneers in leukemia. And so these guys are all in the same field. And so that was what kind of helped me to guide me step by step. And I can say, without beyond a shadow of a doubt, that I've been incredibly blessed to have some of the best possible mentors. And my one advice to you guys, where you guys are in your early stage of trying to find your path, finding your career, finding what fulfills you, is to, number one, always keep your eyes peeled for these mentors. And some of you may not ultimately end up going into biomedical research. Some of you may not be able to go into the scientific field at all. But if, no matter where you go into, there are opportunities to find mentors. And I hope you always seek those guys out, those people who are directly invested in wanting to see you succeed and seeing an aspect of yourself that you might not necessarily see for yourself, and give you a perspective about you and say, look, I think you might be good at this. Maybe you should try it out. People who are willing to take you under your wing. And so I just can't speak highly enough about how critical each step along the way these mentors played in my career development and allowing me to be able to arrive here doing what I love and find fulfilling as a physician scientist doing research in leukemia biology. So what about these blood cancers? So first off, your blood forming cells, they reside in your bone marrow. So they're the bones of your body, the skeletal system. They not only provide you the structure and protection, as you know, but in the middle are the marrows. These are your blood forming cells. This is the factory of your blood forming cells. And they go through this hematopoietic hierarchy, this cascade of differentiation, going from undifferentiated to fully differentiated to produce a whole bunch of different cells that carry on a whole variety of different functions. But at the most primitive level, you have the stem cell, and people have done really elegant studies to show that just transplanting one of these single hematopoietic stem cells from one mouse to another can fully reconstitute the entire hematopoietic hierarchy in that given mouse. And in the middle, you have these progenitor cells. These are cells that kind of divide out from these stem cells and are ready to kind of give out the right output in response to the right stress. So for example, if you were to have a bleeding event and all of a sudden you needed to make more red blood cells, this particular progenitor cell would get revved up. And so there's this whole feedback system that regulates the production of all of these blood forming cells, each of which have different half-lives. So the red blood cell stays in your body typically for about 120 days, whereas the granulocyte lives in your body for about 90 hours before it turns over. So all of these cells have different needs and different requirements, and this is a system to kind of regulate that, but also to have some level of a unifying cell that can give rise to all of these is a very powerful system to reconstitute your blood forming cells and your immune cells. So your red blood cells are here. These are the ones that carry oxygen to your tissues. Your platelets are here. These are the ones that help you clot if you nick yourself while you're shaving. The granulocytes or the neutrophils, these are the bacteria fighting cells that they swallow up the bacteria. If you get a cut and it gets infected, the pus that you see, those are dead neutrophils that have accumulated at that particular site. The macrophages and dendritic cells, they help orchestrate the immune response. They help flag and say, hey, look, I got an infected something here. To these guys, they talk to these guys, the T cells, B cells, and natural killer cells that have the T cells and B cells can specifically recognize certain things. And those are the ones that get, if you get a flu vaccine, those are the ones that get revved up and then also have memory so that when you face it again, you can fight it more effectively. That's the basis of these vaccines. And the natural killer cells are part of your innate immune system where they can fight off cancers and fight off virus-infected cells, not necessarily by specifically recognizing something, but recognizing specific patterns on a given cell. And at every single step of this hematopoietic hierarchy, blood cancers can arise. If you go in the more primitive state, if you look at the stem cell, you can have cancers of the stems that involve the stem cell, like myelodysplastic syndrome, which I'll talk a little bit about, but it's basically a syndrome, a blood syndrome where these stem cells gain mutations and causes them to be ineffective in producing these progeny of blood cells. And not only on top of that, they have a propensity to transform into more aggressive leukemias, such as the acute myeloid leukemia here, and can cause trouble by transforming as well. Further down, you can also have these acute myeloid leukemias. So then these are where the either progenitor cells or stem cells, they're blocked in their ability to differentiate or mature. And so when they're blocked, they're stuck as a baby or a teenager, but can't get to a full functioning adult mature blood cell. And so they crowd out those other normal blood forming cells, causing you to get sick. And I'll talk about the clinical sequelae of why you get sick. So you have acute myeloid leukemia, acute lymphoblastic leukemia. And the major difference between that and these blood cancers here is this differentiation. These leukemias and myelodysplastic syndromes, there's a block in differentiation. Whereas you can get some cells that are blood cancers that are more differentiated. You can have an increase of red blood cells, you can have an increase of platelets, and you can have an increase of granulocytes that's associated with this leukemia called CML, or chronic myelogenous leukemia. You can have an outgrowth of mature dendritic cells, and that's a blood cancer called Langerhans cell histiocytosis. And you can have lymphomas and myelomas that are cancers of the B cells, T cells, and NK cells of various flavors. But the major difference between these blood cancers here and these blood cancers here is that differentiation block. These guys are stuck in differentiating, whereas these guys are mature but continue to grow and also cause trouble in the lymph nodes and the bone marrow and other places as well. And just to simplify things even further, I kind of break this down into a 2x2 matrix. The acute leukemias versus the chronic leukemias, and the lymphoid leukemias versus the myeloid leukemias. So the acute and chronic, you probably already know. Acute just sounds like, these are leukemias, these are blood cancers that come on and hit you clinically very rapidly. These patients feel sick very quickly. They get profoundly sick. They can get life-threatening infections, life-threatening bleeding events, or feel profoundly tired because their oxygen-carrying red blood cells are so low that they can't even, walk a single block, whereas they were running marathons before. And the other part of this is the lymphoid versus myeloid, and those are just the classes of cells here. So your T, natural killer, and B cells are your lymphoid cells, whereas your myeloid cells are these cells, the granulocytes, macrophages, and dendritic cells. So that's just generally how you break them down. Again, acute leukemias, you get very sick very fast. Chronic leukemias, these are much more indolent. A lot of times patients get these, just find out incidentally that they might have these leukemias by getting a blood check at the doctor's office. And Ross talked a little bit about Monday about the stepwise process of how most cancers form. And that was the pioneering work of Bert Vogelstein and Ken Kinsler and their discovery in the context of colorectal cancer, that there's this mutation 1, mutation 2, mutation 3 kind of over time, this sequential acquisition of these mutations that causes a full-blown leukemia, a full-blown colon cancer. Well, the same thing happens for blood cancers, and I'm going to use acute myeloid leukemia as an example. So all of us as healthy adults are walking around with about 10 to 15,000 hematopoietic stem cells. I'm talking about these guys here, these primitive hematopoietic stem cells in your bone marrow. These guys are always spitting out more mature cells to give rise to normal blood-forming cells, but they're also self-renewing to maintain that constant pool of about 10 to 15,000 cells. And every once in a while, a month or so, they'll divide. So, and every time they divide, they can get a mutation. So these hematopoietic stem cells get a mutation on the order of about a dozen a year or once per month. And most of the time, they're completely innocuous. They don't really do anything to you, to the leukemia, to the stem cell. But out of sheer bad luck, sometimes you get a mutation in what happens to be a leukemia-associated gene, but in and of itself does not give rise to a leukemia, but gives this particular leukemia mutation-bearing cell an advantage. So it can nudge out some of these other cells, but it's not a full-blown leukemia. But then within this particular clone that has this mutation, it shuffles the deck again and gets another hit. And there becomes the nidus for a full-blown leukemia. And so there is the establishment of acute myeloid leukemia. Because of a cooperation between this initial mutation and the second hit, that second hit then makes this a full-blown leukemia, which we call the founding clone. And then further after that, it further shuffles the deck and can get additional mutations, or what we call subclones. And they evolve over time so that you can get a whole variety of different subclones with a different constellation of mutations that give rise to the full clinical syndrome or presentation of AML. And these are just a couple of examples of the genes that you can acquire in a sequential fashion that frequently co-occur together as an example of that. But I want to go back to this initial step, this period where you can get these smaller mutations, this pre-leukemic step, because a lot has been learned about this entity that's called clonal hematopoiesis. It's called a whole bunch of different other things, but we like to use the term clonal hematopoiesis. You'll hear all these other terms like CHIP, clonal hematopoiesis of indeterminate potential, age-related clonal hematopoiesis, clonal cytopenias of undetermined significance. All of these kind of mean essentially the same thing with a little bit of nuanced differences across the board, but I'll just refer to this as clonal hematopoiesis. This is what we call a pre-leukemic state. These patients are walking around completely normally, and they actually, were sequenced and found to have a leukemia-associated mutation, but they did not have evidence of a full-blown leukemia. And as you would expect, the older you are, the more of these leukemia-associated mutations occur, if this is more kind of like a randomly occurring thing. And multiple groups have shown this. And not so surprisingly, there's an increased risk of getting a blood cancer, but not as high as you would expect, but definitely there, on the order of about 1% increased risk of getting a leukemia per year, compared to those patients that don't have a clonal hematopoiesis-associated mutation. But what was really striking from the work that these guys did from their research was that these patients had about a 4-fold risk of dying from cardiovascular events, clots within the cardiovascular system in terms of the coronary arteries, or clots that travel to the lung, or clots that travel to the brain to cause strokes. And so it made a lot of people wonder, why was there this association between a leukemia-associated mutation? These patients don't have a full-blown leukemia, but why are they getting clots? And so this clinical correlation led to the need to try to address this using more sophisticated animal models to try to get at causation. So this initial clinical link was then tested in the experimental setting in mice. And so you guys probably already know that when a kind of a clot forms around a coronary vessel, or when a coronary vessel gets blocked off, it's because of cholesterol deposits and calcifications. Calcium gets deposited here and it causes it to get rough and thicker. But these macrophages, these immune cells, actually also come here, and they cause a lot of inflammation. And where do those macrophages come from? They come from myeloid progenitors, and they come from hematopoietic stem cells within the bone marrow. And so then they hypothesized that these leukemia-associated mutations might cause more inflammation, leading to more of these more aggressive and more rapid development of these narrowings of the arteries that could potentially predispose people to get heart attacks, strokes, and clots to the lung. And sure enough, that's what they found when they took mice that had wild-type were reconstituted with wild-type blood cells. They had this size of this is the looking on to the tube of their blood vessels, and so that was the size of the blood vessels. But then the mice that had the leukemia-associated mutations actually had a much larger plaque lesions or plaque size. And so that was the causative link, and suggesting, and so now together with diabetes and high cholesterol, having this kind of pre-leukemic clonal hematopoiesis state might actually be, or is known to be, a independent predictor risk factor for getting cardiovascular disease. So it's an important thing. And so now we want to know, can we intervene on these patients? And so these are important questions that are being asked, and clinical trials and kind of longitudinal following of these patients is being done as we speak. So how do these blood cancers actually cause people to be sick? The main things are The normal blood cells are, as I mentioned, your white blood cells that help fight infection, your red blood cells that carry oxygen to your tissues, and your platelets that help you clot. If you want to sound fancy from using medical terms, you can use these Latin terms. So cytosis means too much of, and cytopenia means deficiency. And so leukocytes is a fancy word for white blood cells. So leukocytosis means too many white blood cells. In those cases, when the leukemia is high and it's growing very rapidly, it can cause clots. It can cause sludging of the blood and slowing down of the blood and cause strokes and heart attacks and difficulty breathing. But leukemias and blood cancers can also cause what's called a deficiency of these cells because they're blocking the normal production of these cells. And you can get what's called leukocytopenias or a deficiency of white blood cells. Because they're blocking the production of normal blood cells, they can also be, your red blood cells can be low. So these are the ones that carry oxygen. And so that because of that, you can feel tired, can get heart attacks because your heart is deprived of oxygen, and have low platelets. And so if you have a deficiency of platelets, you can have abnormal bleeding and you can get rash. This is a classic rash that's associated with low platelet count called petechiae. And then here's just an example of pallor. So if you look at your creases in your hand, they should always be red. If they're not red, and you see this with what's called pallor of the palmar creases, it's actually a very good sensitive marker that your hemoglobin is less than 7, which is very low and probably in need of a transfusion. But these are the kind of clinical manifestations of these acute leukemias and some of these other blood leukemias. Myeloproliferative neoplasms, or MPNs, you can have high blood cell counts. There's 3 or 4 different flavors of MPN. You can have high white blood cell counts and high red blood cell counts, and you can have high platelet counts and have different various clinical manifestations of these. High platelet counts can predispose you to clotting. So this is an example of an angry, inflamed leg that has probably what's called a DVT, or deep vein thrombosis. It's a clot in the deep veins of the leg. And then you can have pancytopenia. So pan means kind of across, and across the board deficiency of these blood forming cells, which for MDS you can have low white blood cell counts, low red blood cell counts, and low platelets. MDS being the myelodysplastic syndrome, that stem cell disorder that I was talking about with ineffective blood forming cells. And beyond the blood forming system, you can have a lot of neurological damage. So you can have increased viscosity. There's one type of blood cancer called Waldenstrom's Macroglobulinemia, where it produces tons and tons of this antibody protein that clogs up the blood and causes your blood to get really viscous and causes clots and confusion and neurological toxicity. Bleeding into the brain, as I mentioned. Damage to the kidneys because of protein deposition in the kidneys, or high abnormal electrolyte abnormalities in potassium that can cause heart rhythm problems. Or you can actually, these leukemias and blood cancers can also cause kind of same thing as like what solid tumors do is outside of the bone marrow, they can also cause tumors in various places and cause blockages of the GI tract or the urinary system or their airways and cause trouble that way. And so how do we as physicians diagnose these blood cancers? Well, it's always trying to take in a full picture. So we do a fair amount of detective work, trying to first take a thorough history, tell us about the symptoms that you're having. A lot of times these patients will have fatigue, they'll have weird infections, fungal infections. Many of us, those of us with normal immune systems should, for the most part, be able to fight off normal fungus. There's fungus and mold everywhere in the air. And so most of the time, if you have a normal immune system, you should not be worried about fungal infections. But if a patient comes in and presents with a fungal infection, that tips you off that raises a red flag for you that you need to be concerned and think about that. Abnormal gums bleeding. Then you examine the patient. If they have swollen gums, sometimes the leukemia can present in gums and cause them to be swollen. Swollen lymph nodes. You look at the skin for rash and look for pallor. Do a cardiac exam. Sometimes if they're anemic, you can hear kind of a murmur in their heart from their heart sounds. You take their blood, you do urine studies to look at protein in the urine, you look at their electrolytes, and you do their blood counts. You do imaging if warranted. You can do a CAT scan, a CT scan, which takes kind of multiple slices of x-rays throughout a given patient's body, or you can do a PET scan, or a positron emission tomography scan, where you inject them with a radioactive sugar and you look at those hypermetabolic cells that are more hungry and want to take up sugar because they're more rapidly growing than normal tissue. You can actually look for spots that are light up on a PET scan as markers of potentially cancer sites of cancer. And then you look under the microscope. So you take a little bit of tissue from their bone marrow, and you also can take their blood and smear it onto a glass slide and look under the microscope. And so this is particularly informative for us as blood cancer doctors. Here's an example of a normal peripheral blood smear. All of these donut red cells that you see, these are the normal shaped red blood cells, and you have some neutrophils here that are dotting this area. This is completely normal. But in myeloma, because myeloma is a cancer of the B cells that produce antibody, you can get what's called this rouleau formation, these kind of red blood cells that stack up on each other like coins. So that tips you off that these proteins are making these red blood cells kind of stick together a little bit more. And here's an example of an abnormal plasma cell. And here's another example of an abnormal cell, what's called a hairy cell leukemia, because of these little hairy projections on here. It's gained the name of what's called hairy cell leukemia. It's a B cell lymphoproliferative disorder, a B cell blood cancer. Here's an example of some bone marrow biopsy slides. So we take a patient, we ask them to lie on their belly, we numb the area behind their hip right here, and then we take a needle and we take off a little bit of the liquid and put that onto a glass slide. And so here is what it looks like. The white here are fat droplets, fat globules. It's completely normal, part of the bone marrow. And then you should see these various different types of blood cells that give rise to all three of the lineages that I was talking about, the whites, the reds, and the platelets. But then when you start seeing kind of uniformity, these sheets of cells, these are leukemic blasts. They have these nuclei that are kind of folded over the chromatin here inside the nuclei. They're a little bit lacy. So this is an example of an abnormal leukemia biopsy. And then here's an example of a myeloma, that B cell blood cancer that I was talking about. So this by looking into the glass slide and discussing with the pathologist, is extremely informative for us to really pinpoint the diagnosis. But there's much more sophisticated studies to do that, to further pinpoint this. So you can see the layers of clinical data that physician scientists go through to try to tease out and nail down a diagnosis, oftentimes which is not very easy to do. Ross, in his lecture, talked about the cytogenetics and kind of genomic changes that happens, and that was initially discovered in the 1960s by Noel and Hungerford at UPenn and Janet Rowley at the University of Chicago. And, you know, at this point, you guys in high school probably are already talking about this next generation sequencing, you know, massively parallel sequencing, where you get down to the very nitty-gritty C's, G's, T's, and A's. But back in the 60s and 70s, this is still also very important genomic interrogation, and this is kind of genomics at lower resolution, but also in particularly something that we still use to diagnose these blood cancers, as we look for pieces of DNA that are swapped in and out for each other to look for patterns of the chromosome changing. Here's an example of what's called a technology called FISH, where we actually look specifically for a region of DNA where we take a fluorescent dye, stick it to a DNA, and allow that DNA to stick to a specific sequence. And they should, for the most part, using these probes, you can kind of label different chromosomes. But if they come together like this, where you have a green and red dot that overlay together, that tells you that two different pieces of DNA from different chromosomes are coming together. This is an abnormality that needs to be worked up. And so that's a technology called FISH that can be used. And then flow cytometry, where you can take individual cells, bone marrow cells, and through single file stain them with fluorescent labeled antibodies or tags, and then look under pass them through this a light beam that can allow these cells to glow various different colors to tell you what other cell surface markers or tags are on the surface of these cells, and how many of them are there that have this tag versus that tag. This is a very useful technology for that. So all of these technologies are being used to pinpoint that diagnosis. So now, for the rest of my time, I'll talk a little bit about treatment of these blood cancers. And think about it largely in about 5 different classes of treatment modalities or treatment approaches. Strikingly enough, you would think, you would be surprised, I guess, that you can treat some blood cancers with even antibiotics. So this is an example of what's called a maltoma. MALT stands for mucosal associated lymphoid tissue. This is the lining of the stomach. And this rough stuff here is what you would see as part of a maltoma. And it's associated with this bacterial infection called H. pylori, or Helicobacter pylori, which is a bacteria when infected in the stomach causes stomach ulcers, and that chronic inflammation can cause this lymphoid tissue that lines the stomach to proliferate and to grow abnormally, and they can cause a maltoma, a cancer. And interestingly enough, if you treat with antibiotics to clear this, the bacteria, you can actually cause these, if you treat it early enough, can cause this particular maltoma to go away. So not typical for most blood cancers, but there's an example by which you can use antibiotics to treat one particular type of blood cancer. More commonly, you guys probably know about chemotherapy. So chemotherapy is just chemical therapy that is, in general, tends to be much more toxic because it's hitting a whole bunch of things. It's hitting not just the cancer cells, but it's hitting a lot of other cells in your body that are growing quickly. So they do this through different mechanisms, by damaging DNA, by depriving them or starving them of nutrients like folic acid or asparagine. Interestingly enough, some acute lymphoblastic leukemias, they can't make their own asparagine out of all the amino acids. And so one treatment approach is to actually give them asparaginase, which is an enzyme that chews up all the asparagine in the circulating system, which the rest of your normal blood cells can make their own asparagine on their own and be just fine. But by depriving specifically these leukemic ALL or lymphoblastic leukemia cells of asparagine is one way to starve them of an essential nutrient that the leukemia specifically needs to grow. There are other poisons that hit the cell division machinery, in particular those microtubules that help pull different cells apart during mitosis. But the name of the game with chemotherapy, is with a bunch of other kind of antibiotics, or in treating tuberculosis or treating HIV, we know that if you treat cancer long enough with just one single modality, one single pathway, if you only just starve them of a particular nutrient, they find ways to get around it. So the way to get around that resistance is to do combination regimens. And so then you're adding one chemotherapy on top of another, and it becomes toxic to the patients, but it also with the hope of trying to kill as much as possible those more rapidly growing cancer cells. So that's chemotherapy. And why is chemotherapy so intensive? The analogy that I use for our patients with acute myeloid leukemia is they start off on day 0 with a diagnosis of acute myeloid leukemia. They have weeds all in their bone marrow. So their bone marrow is packed with these leukemia cells. And when we start induction chemotherapy, it essentially, by day 14, they're in what's called nadir. The weeds are hopefully all gone. Sometimes they're not, but we hope that that garden bed is completely empty. And during this time is when they're most vulnerable to a lot of things. One, they can't make their own red blood cells, so they need to be transfused. They can't make their own platelets, so they need to be transfused that. We can't transfuse infection fighting, the white blood cells, so then we give them antibiotics, but even then they spike fevers and get infections. And so that's why it's so intensive. These patients that are getting this induction chemotherapy, they're with us in the hospital for about a month while we're supporting them and managing their infections and giving them transfusions essentially on a day-to-day basis. And then finally, around day 30 or so, we do another bone marrow biopsy, and our hope is that by then that the weeds have gone away and the flowers have grown back. And so and it's reconstituted with normal blood forming cells, and their normal blood counts come back, and that they're no longer needing transfusions and no longer needing antibiotics because they can fight off the infection. And so that's what we call a remission after they recover their blood counts during this initial time when the garden bed is completely empty. Other treatment approaches is in this class of, I guess I would call it, also lumped in with targeted therapy, but specifically I should make mention of this, is differentiation therapy. So as I mentioned, chemotherapy, the goal is to kill the weeds and hope that the flowers grow back. There is another type of therapy using a targeted therapy called differentiation therapy, where you pinpoint a specific vulnerability of a particular cancer, and instead of wiping away everything and hoping that the flowers grow back, you actually turn the weeds into flowers. And there are a couple of examples of leukemias or blood cancers that do this. One is a specific type of leukemia called acute promyelocytic leukemia. And it is caused by this gene fusion between this gene, PML, or promyelocyte leukemia, and RAR alpha, which stands for retinoic acid receptor alpha. And this is the driving mutation that causes the leukemia. And you can cause very robust clinical responses, remissions, by giving them this drug called ATRA, all-trans-retinoic acid, which is the ligand for this receptor. And ATRA is also this medication called tretinoin, which is actually used to treat acne. And so here's an example of a non-chemotherapeutic drug that has very strong activity and inducing very high remission rates in patients with this specific type of leukemia. Similarly, Ross talked a little bit about these IDH1 and IDH2 mutations in AML. We now have FDA-approved drugs that inhibit IDH1 and IDH2 that similarly turns these IDH mutant leukemias from the weeds into flowers, and that's been a very exciting field of research for us. And then you guys know about the poster child of these other targeted therapies. I won't talk too much about imatinib or Gleevec or BCR-ABL because it's been talked about so much, but this is our goal for all of our blood cancers. Can we really deliver a smart bomb that is specific to the leukemias that's going to be really effective and spare us the toxicity against our normal cells? There's a lot of excitement about other types of targeted therapies. For example, this drug called venetoclax, which targets this specific molecule called BCL2. This is a molecule that is involved in this apoptosis pathway, which is apoptosis is a process by which cells decide to die or commit suicide. And the decision to die, the machinery for that resides within your mitochondria. And you can envision this as a seesaw about whether or not a cell tips over into going into apoptosis. And it's decided by the balance between these pro-death molecules and these anti-death molecules. And BCL2 is one of these anti-death molecules. And cancer cells have a lot of these anti-death molecules, and they prevent themselves from dying. And what venetoclax does is blocks that and prevents them from and shuts down that anti-cell death pathway and basically sweeps the rug out from under it and causing those cells to tip over into committing suicide. And so this has applications for a number of leukemias, including CLL, chronic lymphocytic leukemia, and acute myeloid leukemia. And I'll just say that, the past 5 years have been extremely exciting in this era of targeted therapy. From 1970s to 2017, there's only been a handful of treatments that have been approved and really changed the landscape of therapy for leukemia, blood cancers. But in the past several years, there's just been an explosion. And I won't talk about all these, but many of these are these targeted therapies that fall into this class. The other exciting avenue of research, of course, is immunotherapy, and that's also been all the rage over the past 5 years. One of the mainstays of immunotherapy, one of the first living drugs that we have been giving for our blood cancer patients is something that's been going on for decades now, and that is this concept called allogeneic stem cell transplantation. That means we take blood stem cells from one donor, and before we were going into the donor and doing a bone marrow biopsy, but now we can give a drug, and the stem cells kind of home out of the blood, and we collect them just by sticking a little needle into their vein and collecting those stem cells. And then when a leukemia or blood cancer patient, it gets into as deep of a remission as possible, gets infused these foreign stem cells from the donor, and these stem cells then reconstitutes all the blood-forming cells of this particular patient. So it does two things. It, one, reconstitutes their blood-forming cells, but two, it uses the slight mismatch between this donor and this patient. It uses that slight mismatch to allow those lymphocytes there to recognize, to go and also fight the leukemia, to fight the cancer. And so that's called the graft versus leukemia effect. The graft are those stem cells that take hold in the recipient host. And the leukemia, obviously, that graft is going on to go fight those leukemia cells. Now, it's a double-edged sword because that graft, because of that slight tissue mismatch, can also go cause damage to other sites. For example, the liver, the skin, the lungs, and you can get what's called graft-versus-host disease. So the double-edged sword of of bone marrow transplantation is, the plus is obviously you get T-cell memory. You're giving a living drug that goes and fights this leukemia, and they establish memory and reconstitutes their blood system. But the side effect, and there's a real risk of that, is graft-versus-host disease that can cause this graft to go and reject or fight off the own patient's normal tissues. And that's trying to separate the graft versus leukemia effect and graft-versus-host disease has been something that physician scientists are continuing to do and continuing to improve to do over the years. And so with that, I'll take one moment just to say that I highly, highly encourage all of you to go to bethematch.org if you haven't already, to register to be a bone marrow transplant donor. You have the potential to be a cure, the reason for a cure for a patient with blood cancer. And it's very simple. If you go here and register, they'll mail you a cheek swab. You send in the cheek swab, and they can be in contact with you if you happen to be a match. So that'll be my one plug for bethematch.org. And then I sent to you guys, I don't know if you guys got it, but these links to this YouTube videos about CAR T cells, this idea of chimeric antigen receptor T cells, where a patient with a blood cancer gets collected their T cells that are typically recognizing a whole variety of different things to fight, viruses, cancers, or whatever, recognizing foreign things to attack. But then by infecting it with a virus, completely re-educating all of these T cells to fight off that specific marker on a given leukemia. And they, by doing so, then they infuse this T cell that's been fully re-educated and putting it back into the patient. And you saw the video, or I hope you see the video, of Emily Whitehead, who was the first recipient of this CAR T cell therapy, and is now FDA approved for the treatment of acute lymphoblastic leukemia and a certain type of large cell lymphoma. But her story was truly an inspiration because there were no other therapies that were there for her. She was brave enough to enroll on a clinical trial, and in this clinical trial, she got infused these CAR T cells. She got very sick from it because these T cells caused a huge inflammatory response. She was in the ICU. She had a fever, but then afterward, you hear these doctors talk about it. There was this calm after the storm, and they did a bone marrow biopsy, and she was in complete remission. And these CAR T cells have memory; they remember the antigen, and you hope that they continue to fight off. And in the case of Emily Whitehead, she remains in a remission to this day, and she's the guest of honor at many of these CAR T cell symposium. And so it's a real success story for the field, and a very exciting time for immunotherapy. There are other therapies that are now very exciting on the verge, what's called BiTEs, bispecific T-cell engagers. And similar to CAR T-cells, it's trying to direct T-cells to fight a tumor. And rather than like CAR T-cells, where you infuse and re-educate a T-cell, take a T-cell out of the body and put it back in, you just put a direct link where you try to tie this guy to this guy and allow them to grow and expand and fight off. And so this has been a success story also for acute lymphoblastic leukemia. So I hope that gives you a little flavor of these blood cancers. I'll tell you a little bit about these obstacles to a cure. Obviously, drug resistance is a major problem. In chemotherapy, drug toxicity remains a problem. We need to make sure that we hit the right cells that we need to hit. Clonal heterogeneity, the idea that many of these leukemias can shuffle the deck multiple times and you have different individual ones. And so it's kind of like a game of playing whack-a-mole, where you use one targeted therapy and you get one guy, but there's another guy that comes in and grows out because of the selection pressure that you put on it. And similarly, mutational heterogeneity, so you have different mutations, and so you might not be able to deliver one smart bomb that solves all the problems. And this concept of minimal residual disease, that there's still always this needle in the haystack, that no matter how strong of these therapies that you give, sometimes they can, that there still might be that residual needle in the haystack, and that's all you need for a leukemia cell or a blood cancer cell to grow back. So what are the ways that we can approach or to overcome this research to solve these problems? Mouse modeling, we are now doing injecting directly patient samples directly into mice and giving them blood cancers. Clinical trials is really a mainstay of that. We need more computational power. We need to be able to leverage all the big data sets and use machine learning. And this is how it tells you that I don't know anything about coding, because I have something from the matrix. We are using things like organoids that allows you to do in vitro culture that recapitulates kind of the organ's structures or the integrity of a certain organ, and you can test certain mutations and manipulate that using CRISPR. And chemical biology, using specific chemical probes to instruct a cell to drug proteins or destroy proteins that otherwise doesn't have any enzymatic activity that you can inhibit with a small molecule. So that's, and that's just my ad for you guys, that there's what we need in research and improving the lives of our patients with blood cancers is diversity. We need different ideas, different strategies, and so we need more bright brains like you guys to come into the field, and we're excited for you to be a part of that. So with that, I'll end and take any questions.
Okay, great. Thank you so much, Dr. Kai. This is a wonderful, wonderful, wonderful talk. So we have a couple of questions, and just a reminder, for questions that we cannot answer now, we will be sending you a Google form link for you to insert your question, and Dr. Kai will answer them via Twitter. We have gotten a couple of questions regarding your slides. Will they be available?
Yep, I'll send them out.
Students? Okay, great. Thank you. So the next question we have, Dr. Kai, is what genes are associated with acute and chronic leukemia, if any?
There's a lot, but it's strikingly enough, if you compare them to, say, melanoma or lung cancer, those are kind of where they're associated with a known mutagen, a known cancer-causing agent. With melanoma, sun exposure; with lung cancer, smoking. There's a whole bunch of mutations that are associated with it. With leukemias, acute or chronic, they tend to be a little bit more bland, but there's on average about a dozen or so mutations. Some of the more common ones across all the board of cancers, for example, is this p53 gene. This is a molecule that's one of the most mutated genes across all of cancers, and that is kind of a guardian of the genome. So when there's a DNA damage event, p53 is the gene that says, hold on, we got to stop growing or commit suicide or do something before this gets out of hand. And when that gets mutated, that guardrail, that break is gone, and can cause a number of different leukemias. But there's a handful of other leukemias that all do a handful of other mutations that cause leukemia that do a whole bunch of different things.
Okay, great. Emily Valera asks, isn't CAR T-cell therapy very similar to the plasma therapy that is being used for COVID-19?
Oh, good question. They're different. So they're using two different arms of lymphocytes. So they're both using lymphocytes. The convalescent plasma for COVID-19 is actually using the antibodies that are produced by the B cells, but they're using the proteins, the antibodies that neutralize the virus, and using that to infuse into patients. But the CAR T cells, you're actually taking a cell and infusing that in. The convalescent plasma using the protein that's produced by the cell, whereas in CAR T cells, you're actually infusing a living cell back into the patient that has been re-educated to go and kill the tumor.
Great. And L. Stearns asks, how does the match process work for bone marrow donors? Does blood type indicate a match, or is there another process needed?
There are a number of other matching processes. Largely, it's similar to organ matching or organ transplantation. There's what's called the major antigens. So HLA major antigens. There's class 1, class 2, and then there's minor antigens. But there are these cell surface molecules that are unique to people, and you have about 12 or so of those genes, and you want to make sure that you get as many matches as possible out of those 12. And so then you try to aim for 12 or 10 or 8, but then there's an algorithm for that. And if you get too low and you don't have enough matches, then they go to potentially umbilical cord blood, where you can tolerate a little bit more mismatch. But yes, there's this kind of 12 out of 12 scale of matching system that they look for bone marrow transplant donors and recipients.
Okay, great. Isabella Kutzer asks, can FISH or karyotyping work for diagnosing other forms of solid/liquid tumors or just blood cancer?
It can be used. I don't know if it's routinely used in solid cancers. I think it is, in particular sarcomas that have in pediatric cases where you can have very commonly gene rearrangements. And so, yeah, it's used very commonly across the board, whether or not it's solid tumors or liquid cancers.
Okay, great. Special Sam asks, what is a progenitor?
Progenitor is just a fancy word for more primitive cell. So a cell before it becomes more mature. So a stem cell is kind of like the most primordial, most original cell. Mature cell is, you know, as far down as you can get. A progenitor, I would say, is somewhere in between.
Okay, great. Juan Desmaras asks, can you explain CAR-NK cell therapy and what are the advantages and disadvantages over CAR T cell therapy?
Good question. So the difference between a T cell and an NK cell is that a T cell really there's a real specific lock and key mechanism for that T cell to go and recognize when it needs to fight. The NK cells are a little bit more forgiving. They don't necessarily have to have a perfect lock and key to go where they need to go fight. And I might be simplifying this a little bit too much because a lot of it is beyond my knowledge, but the NK cells that are being led at places like in Boston and St. Louis are using these basically the same idea. They're infusing these cells, but doesn't need to be as specific. And I don't know if they're otherwise doing something where they're re-educating the NK cells or maybe making them produce another hormone or a cytokine that revs up those NK cells. So there may be some parallels, but the major difference in biology between a T cell and an NK cell is that a T cell really needs to have that lock and key fit. It needs to be antigen specific, whereas an NK cell is a little bit more forgiving.
Okay, great. And another attendee asks, how are you able to penetrate the pelvic bone during the biopsy?
A very long needle and a little bit of pressure.
Okay. And how would imagining and radiation play a role in the diagnosis? Can you talk about the CAT scans, if any are involved?
Yeah, so, you know, sometimes, so for example, CLL, chronic lymphocytic leukemia, very indolent leukemia, oftentimes you don't need to treat them, but sometimes a patient might say they have a leukemia, but they might have an enlarged lymph node in their groin. And they say, well, it's really bothering me. Then in that case, we would get imaging to see if it's causing any potential risk or damage to an organ. Could it potentially be blocking one of the kidneys? Could it potentially be blocking the liver? And if so, we use that CT image to help guide us as to whether or not they need treatment. That CLL patient may have completely normal blood counts, but they have a large lymph node in an area that might be risking organ function. We would then, in that case, go and treat it. And that's where we would use imaging for that.
Great. And Jay Alsinawi asks, how does the severity of the cancer change depending on the level of differentiation of the cells it impacts?
Oh, that's a really great question because that has been a project that I've worked on over the past several years. So it does seem that if the leukemia initiates in a stem cell, in a more primitive stem cell, those stem-like features tends to make them a little bit more stubborn to therapies. Whereas if they are leukemias, that same leukemia-causing mutation is introduced into a more differentiated progenitor cell. Even though it's the same mutation, those leukemias tend to be a little bit more sensitive to the drugs. So it does seem to impact drug sensitivity and drug resistance.
Okay, great. And Andrew Zhang asks, how long can CAR T therapy memory keep patients from future leukemia?
Essentially for life. That's the hope. So in fact, you know, one of the interesting things about CAR T cell therapy is that the antigen, the thing that it goes after, happens to be a mark not only on the leukemia cell, but also on normal B cells, the normal B lymphocytes. And fortunately enough, for the most part, we can afford to live without B cells. And so what doctors do is actually, when we follow CAR T cells for their response and making sure that they have memory of this, is we actually follow these patients and make sure that they continuously have undetectable B cells. If the B cells come back, that means that the T cells have lost their memory and are not doing their job. But for the most part, for the patients that are cured of these leukemias from CAR T cells, we should hope that it's lifelong memory.
Okay, great. So we've got a couple of questions as well, not specifically on your talk, but more so on your professional journey regarding what did you study in college and what did you major in? Did that impact where you are now?
I was kind of bread and butter biology, kind of classic for pre-med. I know, people, there's a whole variety of majors that you can do that you don't, I know there are MD, PhD physician scientists who majored in English, but still we're doing a lot of research in the laboratory and stuff. So there are a whole bunch of different paths, but I was kind of bread and butter biology, and not that it made a big impact, but obviously by taking that coursework, it gave me the exposure and training that I needed in the laboratory. So it complemented my research in the laboratory very well.
Okay, great. Maggie Peng asks, how has COVID-19 impacted cancer treatment, especially for patients who are immunocompromised, such as leukemia patients?
Yeah, it's been a real challenge. I mean, I don't have an outpatient panel of patients, so I only see patients in the hospital, but my colleagues who are taking care of patients, patients who, it has factored the decision-making. So, if rather than trying to give patient an intensive chemotherapy where they have to come into the hospital for it, sometimes we would say under the circumstances, if the opportunity cost is that you would expose them to another life-threatening viral infection, then you may say, well, we'll do less intensive chemotherapy, try not to have you come in as frequently as you need for blood transfusions or blood checks, and try to minimize that time for now while we support you. And then once COVID-19 and once the clinics open back up, which as we're doing now, we can start to kind of ramp up and talk about therapy then. So that it has been a major obstacle for my clinical colleagues who are taking care of patients on a day-to-day basis, largely because of the frequent transfusion needs that patients need, but it does also impact their treatment decision kind of thought process.
Okay, great. And could you talk about the different collaborations? So one question was, do different cancer researchers in different fields / specialties, do they collaborate often? And if you could share some of your collaborations that you're doing as well.
Absolutely. Collaboration is the name of the game. Nowadays, the kind of nature of academic research is that you need to be able to tell a full story. And telling a full story means you need to rely on the expertise of a lot of different colleagues. And it's great at institutions like here where, you have a lot of colleagues with a whole bunch of different expertise. My research project in the lab, working in Ross's lab, required me to kind of understand how cell death, how we can investigate the ability of a cell to undergo apoptosis or commit suicide. And there was an expert at Dana-Farber who happened to be down the hall from Scott Armstrong, and he pioneered this technique. And so we reached out to him and he said, sure, send us your cells and we'll do this part. And it's the collaborative spirit of science. And then there's other scientists who are computational people. So down the hall from us is Dr. Richard Kochi, who is our, a computational biologist. And I went to him and said, here's my data. I don't know how to code, but I need to help analyzing this. And so it is an essential framework for how we do science because it just complements each other so well. And so it's a very team-based approach. And I don't think in this day and age you can get by without collaborating.
Okay, great. So our last question is from Megan Singh. So how do you decide whether or not to do stem cell transfusions? How do you judge its benefits, outweigh the risks, and so forth?
Yeah, that's a really great question. So a lot of it has to do with two major things. What is the nature of the leukemia, and how much do you think that patient can tolerate the intensiveness of a transplant? If the leukemia tends to be a little bit better behaving, meaning that the genetic makeup of that leukemia suggests that it might be a little bit more responsive to chemotherapy, based on that genetic information, we can say, this patient probably does not need a stem cell transplant. If it comes back, we have that as an option. But for the most part, we'll give this patient the induction chemotherapy, and then we'll do what's called consolidation chemotherapy, meaning trying to get that needle in the haystack, wiping it clean of every single microscopic cancer cell. But if it's more high risk, and these are the types like p53 mutant leukemias, these are the ones where you want to say, if you can get them into remission, get them to a transplant as quickly as possible, because you need that that graft versus leukemia effect to really take hold, and you're banking on that to give you that. So it's two major calculations that you have to make.
Okay, great. Thank you so much, Dr. Kai, and thank you all for participating. Just a couple of announcements for those questions that weren't answered. Dr. Kai will be answering them via Twitter. His Twitter handle is @ShengFKai. We'll also email the materials that he shared as well for you to view regarding CAR T-cell therapy. And lastly, our next lecture will be on the 16th with Dr. McCohan Moore. And we thank you all again and enjoy the rest of your day. Thank you again, Dr. Kai.
My pleasure. Take care, everyone. Good luck.
Okay.